Hi, so today I would like to discuss the formation of morphogenetic gradient and boundary, especially in the brain. And this is the part 2 of our effort to describe computational methods and to find actually this computational principle that underline the formation of the brain. And so just to remind you that from cell to body, that starting after fertilization of the egg with cell division, according obviously to where the cells are positioned, they are going to become specialized and form the variety of cell that is needed to create an organism. So this is true in this animal development, in plant development, where you reach specialization to form the final shape body, and it is also exemplified here for the formation of the fetus. And so basically the question we're going to be interested in here is how cells are going to become specialized and know where they are and what is needed for this. The short answer is that you need to have an information about the position. Where are the cells compared to other cells? In order for these cells, as we can see here, to trigger a specific genetic code that will only activate specific genes, so that the cell will have its own specificity. And usually it comes with a group of cells, not just one cell, but a group of cells becomes specific. And so this idea of this construction of life, of cell specialization according to the position, bring us to the question, what defines the position? And we will see this has to do with gradients. And so before going into that direction, let me show you various examples. This is long migration of the brain. You have neurons in the thalamus. They need to know where they are, because they need to generate an extension or migration of the tip of the neuron so that they can go through the corridor and go then to the cortex. If you change these gradients, whatever the name of these gradients, it's not important at this moment, then instead of going that way, they miss the target. And in addition, there is also interesting example of two types of gradients. When they are there, neurons are very well segregated. They are going in two different directions. When they are not, by removing them, whatever the technique, then all these neurons become incorrectly projected. And all of this is associated to wrong wiring of the brain, which has many consequences. Another example is what you see here, which is the patterning of the ganglion cells here into the LGN. And you see that if you have the right or the left and right eyes, there is two axes, this VD and nasal temporal axis, and then they're projecting into a space which is defined here with this anterior and posterior axis. And neurons go specifically and stop not at a specific location, but the pattern here filled in a graded manner. And again, we are interested in what defines these coordinates and how mathematically those paths, as you can see here, can be generated, and why and how the neurons know where to stop, and what is ultimately the error, if we can define an error correctly. And this is another example here, the different steps of commissional axon navigating in the spinal cord. Starting from a given position, they have to cross a specific commissural region, and they are attracted by some gradient, so they know where they are. There is a readout, a way to interpret, which we have no idea in terms of computations what this is doing, and this is what we're interested in. So they can reach this region, arrive to this crossing part, and then cross and continue. So they are first attracted, then they forget the first attractant, and they can move on. So another question of interest, which is related to the gradient, is what defines border. So you see here in different colors, the brain is divided in many different regions, as shown here. You have the auditory cortex, olfactory cortex. I mean, you can see where they are located. And yet, you have very nice boundaries. So associated to this notion of gradient, how with this the cells know where is the boundary and what defines the boundary. So this is something also we would like to discuss here from a computational point of view. So making coordinates by morphogenetic gradient, this is an ancient concept, probably starting with the early work of Turing, but this is the principle that you have a graded expression of the molecules so that you can generate an axis with an increasing concentration or a decreasing concentration, as shown here. This is another example taken from the, I think it's the Drosophila patterning. And you can see that then, associated to the gradient, there is expression of genes only in specific region. And so the mechanism of this is not fully understood, but this is a sort of recapitulation of that diffusion is one of the principal components of this morphogenetic gradient. And so let's start actually with C here. You can have a molecule B that follow a diffusion equation and with a degradation. And so this B will be producing, as you can see here, it will produce a graded expression. And so you could have more specifically a molecule L and a receptor. They can bind to associate. This is how the cell knows. So you have a cell. It has here some receptor on the surface, a molecule, some ligand here is coming. They bound together. And so the possibility is that you can have a graded expression of this LR here, or this LR can be degraded, so you can have a removal, and so this leads to some type of exponential decay. And then there are many variants. You can have diffusion and reversible binding, so you have a ligand, you have a receptor that bind, they create this LR, and then you need for this reaction diffusion. And then what you are interested in is what is the profile of expression of the ligand or the ligand bind to the receptor. And usually, again, this leads to a graded expression. So this is the scheme proposed by Volpert and Kerzberg many years ago. And there are different variants where you can have diffusion, reversible binding, and degradation. Diffusion, reversible binding, reversible internalization, and degradation. Internalization means if you have a cell, once you have a ligand receptors, instead of just disappearing, it can be internalized inside the cell. So ligand multiplied by R. And this is well summarized in this paper, this review paper by Lander in Developmental Cell 2002, where you have these basic equations that you can simulate. We will see this, the result in a few minutes. Usually it's very hard to have analytical solution except in here and other cases where you have an exponential decay of the graded expression. And so this is for sure a way to give an expression for axis, because you know basically the cell know where it is, because this is materialized in this number of LR. So what are we going to be interested in here? How gradients are generated and maintained. So we have briefly discussed this, how boundaries are defined and how stable are the boundary. We would like to address this question from a mathematical point of view. So morphogenetic gradient. So the morphogene is a transcription factor. So this is a molecule that upon entering into the cells can activate a specific gene, and so you get the expression of specific protein which allows the cell to become specialized. Okay. Stability of morphogenetic gradient and boundary formation. So we will use some random perturbation. And also what we are going to study is how you can create a shift in the boundary. So suppose you have a boundary at one place here, X. Can you generate a shift in the boundary in one direction, in the left or in the right? And if we have time, we'll discuss open questions. So morphogenetic gradients, so probably the main ideas are coming from Turing more than 70 years ago. And the phenomenological theory of chemical reaction postulate that a substance called morphogens propagates from cells to control chemical rates, which will influence the anatomical form of the organism, which is basically the idea of specialization. And this is formulated in reaction-diffusion. And here we will present the case of two morphogens gradients in various regimes, and we'll analyze the steady state and the effect of small noise. Okay, so then the idea that a substance can propagate from cell to cell, that is, you have a cell, you generate a molecule, and this molecule can go through the extracellular space, go to these cells, and give the cell the specialty. But of course, if this process is invariant by symmetry, which means that what happened in one is the same as what happened in two, then there will not be an expression of decreasing of increasing. So you need here a mechanism for specifically addressing this question. So before this, let's go to the concept of French flag, which is an idea of positional information which is necessary and sufficient to translate genetic information into patterning. So basically the idea is the following. The cell is able to read this gradient. So you have an expression here of a molecule, and there are thresholds that are not clearly defined, although mathematically we can try, based on chemical reaction, to associate this threshold to, let's say, the combination or the addition of the number of molecules that are externally to the cell, the morphogenetic activity, and the number of binding sites. So if you have more, then not all the binding sites are going to be activated. That could lead to these blue type cells, which means expression of a certain gene. And if you're exactly at the right number of binding sites, maybe then you express another types of proteins. And if there are more, maybe you can express some repressor that will generate another type of main expression. And that's the idea of this French flag patterning, that according to the cell position it expresses, it becomes specialized and different. Okay, so this is this notion of threshold, which is, there are efforts. I will give you an example where we did a model a couple of years ago. So you can go to this reference and paper to see the exact mathematics. Maybe I'll do a YouTube on this. So morphogenetic decreasing concentration of a chemical morphogen is a scenario where cells are labeled and ordered, which is a way to generate a local coordinate system. Okay, so not only the cells become specialized, but according to this, you have here region 1, region 2, and region 3. So you start from an egg by division to have cells that become specialized, although here we do not assume that the cells are moving yet, but still you have a patterning. So we already saw this part. I explained you briefly the different scenarios, and here what you can see is example as a function of the distance of the various expression of the gradient depending on different parameters. And you can have long-range gradient, but also here you can have very short-range gradient where the gradient is very steep. And here you can have also other type of gradient where instead of being very steep, they are almost one, and then decays again in a very steep manner. So this is not that great to have, let's say 1 and 2, to have a good patterning, because you almost see the same thing for a long distance. So you have intermediate cases where here you can have a patterning, and so this allows at least to have a more specific region where you have this sharp transition. So the last idea that I didn't discuss much is the fact that you can have these patternings through transcytosis. So you have the expression in one cell here, and the molecule pass in the other cell, so these cells start to produce, and so on, and so you have a production. And so the diffusion is not just diffusion by itself, but it's a mechanism of diffusion and release and genesis that is done over each cell. And it could be more complicated. It could involve here this receptor ligand interaction, but the idea is, in the end, you generate a gradient that leads to various cell specialization, as we saw here for the case of the French flag. So now the scenario of morphogenetic gradients. So we have seen that it can be achieved by passive diffusion, by this transcytosis, although there are drawback for this transcytosis, because it might be too long to take place. But this is a well-considered scenario. So let's go now to the mathematical model. So I present a scenario based on a transcription factor. So just to remind you, a transcription factor is a molecule that can bind to the DNA so that it can express specific messenger RNA, which express a protein of interest. And they are very specific. They can bind to, very specific region of the DNA. When the chromatin opens, they can reach this region. And of course, all this regulation about opening the chromatin is a subject by itself. So the transcription factor bound to a specific DNA site to generate a specific protein. And so the propagation of morphogen in the model we're going to discuss here, you can generate a molecule that is going to be secreted. It passes through one cell, and if the cell is the same, it basically does the same thing, and again and again, and so you can have this genesis. And here we discuss the case of unidirectional flux, that it can go to one direction. And here we discuss the fact that this molecule can be generated and go to two directions. And why it go to one direction compared to the other, this is an assumption, because here we need a specific biological mechanisms for this. And so this scenario, where this is what we did more or less 15 years ago, and I'm going to describe them now. So you have a DNA molecules, it binds to this transcription factor to produce molecules. This molecule can be killed, hydrolyzed by E, but then when you have this molecule that is generated, so when A crosses the cell membrane, it becomes a little bit modified, which as a consequence reduce the efficiency of the production of A. So when this new molecule, this new transcription factor, which could be modified, and this is an assumption, lead to the production of A, then you have this equation. And so if you have the binding site of the DNA, N plus A, this leads to this ANA star component, and you can use this Michaelis Mentens approximation, and this is the rate N of A star at cell K, which is proportional to NK, but it's also proportional to this AK that is coming from the previous cells. And so this is a way to keep the memory and allow us to have this decay in the expression. So A star may arise from a lipid that is attached when crossing the cell membrane. And so now you can write the equation of production for AK, which is a transcription factor, the morphogenes, which depends on this Michaelis Mentens and AK that we just discussed. There is here a certain degradation rate. And here this is what happened when you propagate. So this is inside one cell, and this is not due to the propagation. So it comes from the cell K-1, and AK can be degraded with a certain rate. And this is the initial cells. And this is a case of a unidirectional propagation. These are changed for a bidirectional, that is when it can go to both directions in a cell. And then for this, you change this flux by adding what happened on the left and what happened on the right. Okay, so this is the different parameters. K is the synthesis rate of A per active chromatin site, controlled by this A star that is coming when the element acquire modification crossing the membrane. Gamma is a hydrolysis rate. Gamma T is a proportion of A packed into vesicle. This is our model here. And phi is the fraction of A entering a cell when experiencing a conformational change to A. And so what you can study here are the steady state solutions when you just write ddt equals 0. So you have then equation of this type to write. And what is very nice is that you can solve this equation by induction, and you see that AK decays like 1 over K. And so you have here a graded expression, and this is the case when alpha is 1. When alpha is not 1, then you have this exponential decay with decay like lambda k with some little modification here. And this is what you get in the case of bidirectionals. You have two modes, lambda plus and lambda minus. And again, you have here this exponential decay rate. So when you do simulations, you can see, interestingly, that you can also from this model get some long-range patterning. When alpha equals 1 and decays, you can see how this is changing. And also you can have for various parameter of alpha some cases where the gradient is shallow. This is a unidirectional case. In the bidirectional case with a zero flux boundary conditions, you can see that boundary condition because you need to see to express what happened for the last cell. But basically you can have long extension where you have almost no variation and then a sharp variation toward the end. So activation with multiple binding sites. So this is what I mentioned earlier. You can look at the probability that K binding sites are occupied when there are a certain number of NF transcription factor, and this is given by a Markov chains. And what we found, and this is, I'm not going to describe much this here, just to mention this, at least 2 binding sites are required to produce here a genetic switch, which allows actually a genetic switch is the possibility that you express something very high in some region and suddenly there's a switch, nothing is expressed in the other region, although you have a graded expression here in yellow. So this is a graded expression. Okay, and this is mentioned here, and I will probably give a specific lecture on this later on. And I'll finish with this by mentioning this computational review. So I will stop now and resume in the next YouTube video for what happened to define the specific boundary between two gradients. Thank you.